Well, it's a real pleasure to be with you virtually. I apologize that I couldn't make the trip in person, but I had unmovable commitments. Um, so as you'll see from the presentation, before I get started, I just wanted to say a word that you'll see that my work and Joe's work have diverged considerably over the years since my graduation. But what I learned from Joe in terms of rigorous thinking skills, formulating a coherent narrative, writing clearly and eloquently, I will never forget. And I'm very, very grateful for having had Joe's tutelage in my early formative years. So thank you, Joe, for everything that you did to help me become a better scientist. So the work that I'll tell you about today is work that we've been doing at where I am today, which is a company called Insitro, which I founded just about 5 years ago. And it is a drug discovery and development company that leverages machine learning to help bring better medicines to patients. So the fundamental premise behind our work in Insitro is that many complex diseases currently remain without treatment because they are opaque and poorly defined. The taxonomy that we use to define them is typically based on coarse subjective clinical labels, often dating back decades, and don't really capture the underlying biological foundations of the disease, which often causes us to lump together diseases that are actually distinct or separate out diseases that are actually the same. And so we believe that in order to uncover new disease modifying drugs, we need to better unravel the underlying disease biology and identify actionable causal nodes. The problem is that human biology is really complicated, far too complicated for the human brain to understand. And so the path that we've taken to get to the goal on the previous slide is to really try and collect a lot of data that is unbiased, that provides multiple complementary perspectives on human biology, and then let machines do what they now do far better than we are able to do, which is interpret those data to find patterns, and those patterns will hopefully help us redefine disease, identify those actionable causal nodes, and hence potentially effective hub for therapeutic interventions. So the kind of approach that we've taken to do that is to collect data across two different types. In the wet labs that we've been building for the past 5 years, we build cellular disease models that take human-derived cellular systems that we'll talk about a little bit later, where we can introduce different types of healthy and diseased genetics, and then interrogate those cells using multiple phenotypic modalities with a focus on modalities that are unbiased, that give us, again, these complementary perspectives on cell state and health and disease. So we have different types of omic modality, transcriptomics, proteomics. We have fixed and live cell imaging, as well as different activity phenotypes that are specific to certain kinds of functional readouts. Interrogating health and disease in cells is wonderful, but ultimately you need to help to intervene not in cells, but in humans. And so the complementary data modality that we've taken is to collect high content data from humans. Here, again, the focus is to try and walk away from the preconceptions that people have about what matters and what doesn't. And so we take large data sets of unbiased data modalities, whether it's biomarkers in blood or tissue, or different kinds of imaging, histopathology, as well as organism level imaging. So with these very large amounts of data that people can't really assimilate, we use machine learning to help unravel the underlying biology of health and disease. And what's important here is, again, we're trying to walk away from what people believe is important or not. So the focus is to use the novel method of self-supervised or representation learning methods, where we feed in all of these data modalities into a self-supervised learning algorithm that gives us a biological state embedding that tells us where people at or cells from people lie on a manifold that spans health and disease. And that much more concise and semantically relevant representation is also much more actionable. So how do we then get to actions or causality? And before that, and that much more precise representation allows us to take this fuzzy notion of disease that we have today and turn it into something that is much more crisp and actionable. So where do actions come from? How do we get to causality in biology, where it's very easy to get lost in confounders and non-causal associations? So for that, the one rare source of ground truth on causality that we have is human genetics. And we leverage genetics in two different ways. In our own cellular disease models, we can actually do interventional experiments. That is, we can intervene in the cells by genetically modifying them and do a direct comparison in what's called an isogenic line between the healthy and the disease genetics to directly measure the causal effect. In humans, of course, you can't really do that. And so in humans, we leverage natural genetics, or a process known in genetics as Mendelian randomization, where we leverage the natural human variation in order to understand that cause-effect relationships between genetics and the human phenotypes. So putting those together, that gives us a set of targets, which are high confidence genetic drivers that we hope will meaningfully modulate clinical outcomes in a patient population that is relevant to that intervention, which is also a population that is defined via machine learning using a biomarker on these high content data sets. And then our goal, of course, is to identify therapeutic interventions, which are molecules that modulate these targets in these patients. So we have three focus areas in terms of where we apply this work. We're working in the areas of metabolism, neuroscience, and oncology, using both of these modalities and their complementary strengths. And what I'll tell you about today is actually two vignettes. One is using clinical data to interrogate oncology, and one is using cellular data to interrogate neuroscience, just to show these two different capabilities. So starting with the work on clinical data in oncology, part of the realization that brought us to oncology is that there's been a lot of very great successes in oncology over the past decade, but it seems to sort of be petering out over the last few years. There's been two main sources of oncology targets that have been very successful. There are precision medicine drivers of disease that are cell autonomous, that occur specifically in tumor cells. And then there are tumor antigens, which are molecules that present, or proteins that present on the cell surface of tumors and allow targeted immunotherapies to be directed specifically towards tumor cells and not towards the healthy tissue that surrounds them. Those have been incredibly successful and have totally transformed the care of cancer patients across multiple different cancer types, but they've kind of ground to a halt a little bit. And partly it's because there's many of the new targets that have emerged as potentially promising haven't really panned out in the clinic for reasons that no one fully understands. And the other is that even the molecules that we've generated that have been very successful only work for a relatively small subset of the patient population, again, for reasons that no one fully understands. So everyone talks about immuno-oncology. In the best of cancers where immuno-oncology works, it's successful for about 30% of the patients. Oftentimes it's more like 10%, and there has not been a lot of success in narrowing down what subsets are the ones that these drugs are going to be effective for, and why. And so there's really a need to interrogate the underlying biologies in order to get both to an understanding of the relevant patient populations, as well as to the next set of targets that might be helpful. So the focus that we've taken here is to really leverage human cohorts, and that's because of the complexity of cancer and the challenges interrogating it in a cell autonomous way in a dish. And so what we've done is we've looked to a number of large human cohorts in which high content data from patients is collected, and then applied machine learning to identify patterns there. I'll tell you about the earliest work that we did in a fairly old cohort called TCGA, the Cancer Genome Atlas, but we've since done that in a number of other cohorts with very significant, interesting findings. So TCGA was one of the first largest collections of cancer data that was generated by the community. It is in some ways a wonderful cohort. It has over 20,000 over 20,000 samples from tumor and normal, over 33 cancer types, with different diverse data modalities that are each informative, but they're also very sporadically collected. And that's because TCGA is actually a mashup of fairly diverse cohorts that were collected each in a different hospital and then put together, which also makes it quite challenging from a machine learning perspective because it's not harmonized or uniform. So in order to work on this cohort and others, we took that approach that I mentioned a few slides ago, which is let's learn the language of clinical data from what is the most readily collected and broadly available data modality, which is also surprisingly information rich. So this is the histopathology, or what's called H&E slides. They're collected as biopsy samples in pretty much every solid tumor patient, almost everywhere in the world. And so there's millions and millions, probably more than that, of these images that have been digitized and are available. So we apply self-supervised deep learning to those images, and that gives us a way of transforming what is an image of billions of pixels into something that is much more concise and semantically relevant, which is only a few hundred dimensions large. And because what we have is so concise and also semantically relevant, we can now overlay on top of it smaller cohorts where we have additional high content data where you wouldn't be powered to find associations if you were to be looking at the original images, but in this representation, you're powered for association as well as for causality. So first, let me show you that there is a tremendous amount of information left in histopathology images that is not currently being leveraged. So what you see here is what's called the Kaplan-Meier curve, which is a survival curve. So the x-axis is years, the y-axis is the fraction of patients that survive over those number of years. And you can see a stratification between high risk and low risk patients, low risk being at the top. And the left-hand curve is what you would get using standard clinical risk covariates that are measured as part of the standard of care. What you see on the right is what happens when you take information that is derived from the self-supervised learning and add it on top of those same covariates. And you can see this is specifically in breast cancer, but we've done this in a number of other cases as well, a much better stratification with a stronger hazard ratio and a better p-value. So that demonstrates that there is considerable information that's in the embedding from histopathology that pathologists are not currently looking at. But the next slide shows just how much of that there is, because what you see on the x-axis is that same clinical risk score that you saw on the left, and on the right-hand side is the additive component that you got from the embedding on the histopathology, and you can see that these are effectively uncorrelated with each other. That is, the R-squared is actually 0, which demonstrates that what the machine learning can see in those images is entirely orthogonal to what a clinician knows to look for. So that's great, but it doesn't actually give us actionable interventions in terms of where do you go in order to turn this into targets. And so in order to get to that, what we did is we took that same embedding, learned in the same self-supervised way, and started to overlay these smaller cohorts where we've measured molecular markers. So first of all, this is mostly a proof of concept. We looked at the notion of tumor somatic mutations that are collected for a subset of patients, those that are typically the better off and are and have access to tertiary care centers. And what we're able to do is show that from the same histology image that's collected from, as I said, every solid tumor patient almost everywhere in the world, you can actually predict with very high accuracy the somatic mutations directly from the histology image, which allows you to kind of in some ways identify which patients are likely to benefit from precision therapeutics without actually having to do the genetics. So that's nice from the perspective of improving standard of care for those patients, but those somatic variants are rarely novel targets. So how do we get to novel targets from this? And to do that, we turned actually, let me go to this to another form of molecular measurement that is much more unbiased and much less trodden. So this is an assay that was developed by a former Stanford colleague. It's called ATAC-seq, and without getting into the detailed biology of it, what it measures is the accessibility of the chromatin, and so that is which regions of the DNA are open versus closed open typically corresponding to active, closed corresponding to not active. And this is a genome-wide measurement that is very informative because it also, in addition to allowing you to understand the level of activity of genes that are transcribed, that is protein coding genes, it also allows you to identify open regions of the chromatin that are not coding regions, that are regulatory regions, and those are often very strongly implicated in cancer as well and are very hard to measure using alternative assays. So this is a really valuable form of measurement that is very cancer relevant, but it's also something that you would never use clinically because it is a very finicky assay. It is something that is very sensitive to how the samples are handled. And so it's really only available in small scale research data sets like the one that you see on the bottom right, which is a very small subset of TCGA that was collected by, where the samples were taken by that same Stanford lab that developed the assay to begin with, and a TAC-seq was measured on those 400 patients. So this is 400 patients spread peanut butter thin over 23 cancer types, which means that the largest subcohort within that set is about 25 to 30 breast cancer patients. And so while it gives you some insight, certainly about chromatin activity within those patients, it's not powered for any meaningful statistical analysis. And so the question that we asked ourselves is, can we take the histopathology, those images that are so readily available, and impute, train a model to impute the attack-seq profile from the histology alone. So we look at the histology and predict open and closed chromatin. And if you could make those predictions, you'd be able to then take any histology sample, including in the context of TCGA, an additional 11,000 patients, and impute the attack-seq signature. So when we first presented this idea to colleagues, including ones who developed the attack-seq assay, they said, yeah, it's not going to work. But it turns out that surprisingly it actually does. So you can look, if you think about the attack-seq profile, there's about 500,000 peaks along the genome that change in their open versus closed status. 5,000 or 1% of those are predictable from the histology alone with an R-squared of greater than or equal to 0.5, using the same embedding that I showed you a few slides ago. That is not something that was uniquely trained to this. And what you see here on the right is the quality of that predictive accuracy. When you see on the left is the actual attack-seq, on the right is the predicted attack-seq. Needless to say, on held out test patients, so you're not overfitting to the training data. And at the bottom right, you see that there is a very strong R-squared between the predicted and the actual, demonstrating that these are very highly predictable. And so now all of a sudden, you have imputed attack-seq not on 400 patients, but on 11,000 patients, and you can start asking really fun and important questions. Like, are there peaks whose openness versus closedness is associated with survival? If you have 30 breast cancer patients, there's not a signal for survival. There's just not enough patients who lived versus died. But in this case, you can actually start asking those questions. And what you can now see here, and I'm assuming most of you are not breast cancer oncology experts, but what we see here is that you actually are able to identify genes whose activity status is highly correlated or associated with survival in breast cancer on the left, and with triple negative breast cancer on the right. Triple negative breast cancer being a disease where the current standard of care is really chemotherapy, and people typically die quite early after diagnosis. And here we find that there are genes whose activity gives us a hazard ratio of 2 or so, and some of those are well-known, characterized genes, and others are novel to triple negative, even though they've been characterized as being tumorigenic in other cancers. And so those are potentially quite interesting novel TNBC targets, which we're now prosecuting in terms of potential therapeutic interventions. An additional form of evidence for those targets comes in this additional form where we go to somatic mutations in those patients, and this is specifically in TNBC. What you see here is looking at the columns are patients, the rows are these attacked targets, and the red are cases where the cancer has mutated to amplify or greatly increase the activity of those genes. And so this demonstrates, just by looking at this chart, that there's a considerable enrichment for amplification events in those genes among those patients, again, providing an orthogonal form of support to the fact that these genes, by their activity, tend to lend selective advantage to the tumor cells. And so this kind of is a picture of how we approach cancer, but we're now applying the same approach in other therapeutic areas. And so here the idea is to take very rich, high content data source that is highly abundant, in this case histopathology, use machine learning to learn the language of that data modality, and then from that impute additional phenotypes that are more actionable than images, which in this case was ATAC-seq, but we're also doing it for our RNA-seq and others. And that reveals genetic targets that could then be the starting point for therapeutic development. We're doing the same thing now for metabolic disease using abdominal MRI, for neurodegenerative disease using brain MRI, and so on. Okay, so shifting gears, I mentioned that we have two sources of data modalities in the work that we do. I'm now going to talk about cellular data. I'm going to use neuroscience as an example for that. So here the idea is that now we're going to the cellular side, and we are using the genetics here as the starting point of engineering disease models that allow us to interrogate disease. So we start out with a healthy cell, what's called an induced pluripotent stem cell. This is something that could then be differentiated into diverse lineages depending on which disease you're looking to study. And then we introduce a disease-causing mutation or more than one into those cells so that you have healthy cells being the wild type and sick being the mutation on top of that healthy. And so you end up with what's called an isogenic set of cells, so that you have a very tightly regulated case control, because the only thing that changed is that disease-causing mutation. We then take that population of cells, and we measure them using a range of different modalities, which gives us a very broad view of cell state. And then we learn the self-supervised embedding on top of those modalities, and then overlay the sick versus healthy on top of that to give us a, quote, disease axis that allows us to understand what the disease does to the cell and potentially what interventions might revert it. So in order to assay cells, we've developed a whole range of different machine learning enabled technologies. This is just one example of such. This is a really cool microscope that we developed in-house that combines optics and machine learning. It uses the fact that when light refracts into these semi-transparent cells, its phase changes. And so in addition to collecting the amplitude of the light, you can also collect the phase. And what we've done here is actually shine light into the cells at different angles, which allows the phase to be collected at different angles. And the machine learning is used to reconstruct a high resolution, high contrast version of the cells in those live cells. On top of that, we layer another layer of machine learning, which actually looks at those cells and imputes specific markers for the cell nucleus, for the cell membrane, for lipid droplets, and so on. And those are trained by taking cells, fixing them, staining them, and then training a machine learning to look at the phase output as input and then impute those biomarker stains. What's important is that although to train the model, you've had to fix and stain the cells, which basically kills them. In real life, having trained the model, we can now do this on top of the live cells. And so you could actually start to track cells in live cell imaging with all of the benefits of the extra information you get from cell staining. And this is just an example of the real versus the predicted. And again, you can see very similar visually results. With that set of modalities, we can now ask questions like, how do you assess at large scale what different genetic interventions do to cells, both in terms of causing disease phenotypes, as well as reverting disease phenotypes? And so we've built a very sophisticated optical screening platform that basically takes a set of cells in a dish and introduces CRISPR guides into them in a pool with an average infectivity of 1. So think about this as a whole mess of genetic perturbations introduced into the cell, where each cell on average gets 1 guide and therefore gets 1 genetic edit. The resulting is a heterogeneous set of cells all in a single well. And now you can go and phenotype those cells using technologies like the live cell imaging that I showed on the previous slide. What you see here in the middle panel is actually an output from that. Those little dots that you're moving are mitochondria moving across axons between one neuron and the other. But you can also use other forms of imaging. And having assayed those cells and measured them in all sorts of different ways, you can now fix them and sequence the barcode that that identifies which guide each cell got, so that you can now associate the genotype that each cell has to the phenotype that was measured at a single cell level, which is an incredible rich capability. So,
Daphne, can you
yes.
kind of start wrapping up?
Yes, okay. So anyway, so we built this, we scaled this, and we applied this to a monogenic disease called tuberous sclerosis complex, which is a pediatric epilepsy, which is a devastating disease. And then we took those, we screened for interventions, and we found a set of targets that basically revert the phenotype that we saw in the dish. And then we tested it on live cells at 50 days. And we showed that those novel targets that we identified reverted the phenotype of an epileptic seizure that you can see in a dish that mirrors the same kind of dysfunction that these epileptic patients manifest in EEG. So I won't show you the data, but it's really quite compelling in terms of the reversion of the epileptic phenotype. We're now applying this to ALS, which is a devastating neurodegenerative disease. And here the genetics are much more complex. It's not just a single genetic variant, but multiple different genetic variants. And so we've built out a very significant library of different genetic variants, and are now identifying phenotypes as well as reversions. And I wasn't planning to show that anyway, because it's work in progress. So maybe just to summarize, what we've built is an engineered discovery process that really puts together data from different modalities, both in vitro data as well as human clinical data, feeding into a uniform data platform interrogated via machine learning that then gives us novel insights about health and disease. And so maybe just to sort of wrap up in one more minute, is that I'm really excited about this direction of work, because I think back on, the history of science and reflect that there were periods in history where a certain scientific discipline moved incredibly quickly for a certain period of time because of a new way of measuring things or a new type of insight. So in the late 1800s, that was chemistry with the discovery of the periodic table. Then in the early 1900s, it was physics with understanding connection between matter and energy. 1950s, of course, computing with the invention of semiconductors. And then in the 1990s, we kind of had these two disciplines starting to evolve in parallel. Quantitative biology, where all of a sudden we were able to measure biology at fidelity and scale in numerical ways. And of course, data science / machine learning. And they kind of evolved independently of each other. But I think where things are going to be in the next 20 years is the era of digital biology, where we're going to be able to measure biology at unprecedented fidelity and scale, interrogate what we see using machine learning methods, and then engineer biology to do things that it would not normally otherwise do. And this will have incredible repercussions, I think, in the area of human health, but also in agriculture and biomaterials and energy and the environment. And I think it's going to be an incredible amount of progress that will hopefully make our world a better place. And with that, I will stop. Thank you.